Sorry, Dr. Adora, if you could please stay online, but just mute yourself for the next flash talks. That would be great, because we'll have a panel Q&A. I'd now like to invite Dr. Edel Hyland for the first flash talk. Thank you.
Hello. Hello, everyone. I have 5 minutes to give you a vignette into what I do here in my lab. So we look at, so Linda introduced us to fungal infections. That's what I work on too. I work specifically on candida, pathogenic yeast. And so everyone now in the fungal community will start their talk with this, because it was the first time the international community recognized the importance of fungal pathogens. It's about 3 years old now. And it's really, really good to see that fungal infections have gotten some recognition. So out of these 11, either critical priority or high priority, 5 of these are causative agents of candidiasis, the disease that I work on. And this, so Linda also showed this, but I really like to show this figure because it really brings home how much they've been overlooked. So in black are the bars for incidence, or the number of patients that have disease, and in yellow are the bars, the percentage that die from this. So you can see malaria, TB, HIV, the black, there's huge incidence of these throughout the world, but these yellow bars are much lower, right? Whereas for all the fungal infections, it's the reverse. The yellow bars are much higher. There's a lot more mortality associated with the disease. And the sad thing about this graph here is it's because it's not that the fungi are much more effective or anything. It's just there hasn't been attention and resources put into the fungal community. And what happens here, these are opportunistic pathogens, right? So we all have candida in our microbiome. It's not problematic, but it's these vulnerable people, the immunocompromised, the ones that are on chemotherapy, the ones that are getting their transplant, they're the ones that become vulnerable. And the medical community has come so far in getting better chemotherapy drugs, better transplant operations, but yet these succumb to fungal infections at the end. And it's really an oversight that's detrimental to these people. Okay, so I work on this one here, Naccomyces glabratus, formerly known as Candida glabratus. This has a high incidence of multi-drug resistance. It's intrinsically resistant to the most basic, the frontline treatment for fungal infections, and that's azoles. So it's a real problem to treat this infection if you get it. I wanted to look to see how prevalent MDR was here, so I work with someone over in the Royal Hospital, Dr. Derek Farley. He gave me 83 non-Candida albicans. Candida albicans is the most prevalent Candida species. It accounts for about 70% of this fungal infection. The next one down is Naccomyces glabratus. So out of the 83 non-Candida albicans isolates we got from ICU patients in the Royal, 61 of them were the species that I work on. And out of them, we asked how many of them could grow at concentrations above the clinical breakpoint of different antifungals. So we tried 3 common antifungals used. We just did a qualitative, could they grow or could they not grow? And we found that there was 41 of the 61 glabrata isolates that could grow above the clinical breakpoints of 3 antifungals. And here's kind of the breakdown of that. Thankfully, there's nothing that's completely resistant to all 3 of these drugs, but you can see there is prevalence of multi-drug resistance here at Belfast. Okay, so what is my lab trying to do to tackle this problem? So we have 3 kind of major themes going in the lab. The first one is we're looking for novel resistance breakers. So antifungal resistance is quite a different beast to antibacterial resistance. The mechanisms are quite different. We can talk about it later. But so what we were looking to find were genes in our species that made Candida glabrata more susceptible to drug. So this is a very simple, this is the only piece of data I will show. The wild type, or the normal strain, when you don't have drug, it grows fine. The dash is a strain that lacks a certain gene. It grows fine without drug. But then when you add in the azole drug, the strain with the dash, the deletion, doesn't grow. So we've identified genes in the species that make them more susceptible to the common azole drug. We did in silico drug screens for the protein products of these genes to identify inhibitors that'll bind and hopefully work. And we have some lead compounds now. We're calling these resistant breakers. So they will work synergistically with the antifungals that are commonly being used in order to kill this yeast. The second theme in the lab is this phenotype called antifungal drug tolerance. So if you're not in the fungal world, you'll kind of think, and you're in the bacterial world, you might be familiar with persister cells. It's basically very similar. It's non-genetic based resistance mechanisms. So if we look at this DDA assay, this disc diffusion assay, where we usually look for drug susceptibility, after 48 hours, you will see cells appear inside the zone of inhibition, inside that little yellow circle, indicating that they can survive the presence of these really high drug concentrations. But this is not resistance. You can take them up and plate them on new cells, and they'll give you the exact same pattern again. It's not genetic, it's phenotypic. So we're using functional genomics and single-cell analysis to really get to the bottom of the mechanism here and see, can we find targets that will treat tolerant cells. And then the last vein in the lab is kind of a more One Health approach to candida drug resistance. We're calling it azole cross-resistance. So I'll just describe, or I'll define for you how we think about cross-resistance. So resistance is a particular mechanism within an organism that prevents it from being killed by a drug. Multi-drug resistance would be two distinct mechanisms in that organism that now make it resistant to two drugs. But cross-resistance would be a single mechanism that makes it resistant to both drugs. And so we're defining this kind of azole cross-resistance as a yeast cell that has resistance to different types of azoles. So another problem with fungi and antifungal resistance are very few drugs, and we use the same drugs in the field as we do in the clinic. And there's a lot of resistance that's generated in the agricultural settings that now ends up in the clinic. And so we have these different drugs in the lab, and we want to see, can we identify these cross-resistant mechanisms whereby yeast can become tolerant or resistant to both clinical and agricultural azoles. And when we just look at the clinical samples we have from Belfast Hospital, we can find that 30% of them do show resistance to these agricultural azoles. So it is prevalent, and it is something that we all should be looking at. And with that, I'll thank you very much for listening.
Thank you.